All right, hi everybody. So I realized that last time we created the FastQC report, but we never actually talked about what some of this stuff means. So I figured go ahead and talk about it today, and then next time we can run the FastQC, or we can run FastQC on the trimmed reads, which we have done now, along with MultiQC on the trimming files to find out some of the statistics for actually trimming, whereas the FastQC will just tell us about the new reads. So we'll be able to compare whether or not the per base sequence content is better after trimming, or if we have less warnings and less failures after trimming than we did beforehand. So in the description down below, I'll also put a link to the FastQC manual. Some things I've noticed are a little out of date on it, and some things aren't included, like some of the summary stuff on the side, like the titles aren't included in it, but it does give a good overview. Personally, I like hearing somebody explain about it, so that's what I'm going to try to do today, versus just reading through a manual. But here we have both the SRR 2121-770 forward read that we had looked at a little bit last time before we went on to trimming. And then we also have the MultiQC report. And the MultiQC report has everything, all of our reads thrown together. So we have our SRR 2121-770 forward, 770 reverse, 771 forward, reverse, so on and so forth. All the way down. We can click this to show everything. So this is just a consolidation of all of our FastQC reports on each read, making it more digestible. But we'll look at this and we can always go back and forth. So the first thing that we see is our basic statistics. So this is just going to give us our file name. So this tells us that we're looking at the 770 forward read. File type, just basic. We have Illumina reads. Sanger, I'll mention this a little bit later when we start talking about the per base end content. Sanger always has a really high per base end content at the beginning and the end, because the first 25 are just bad reads. The total sequences that we have for all of the forward is going to be a little over 818 million. So 118,323,219. Sequences flagged for poor quality. I'm not 100% sure what is considered poor quality here, but we have 0. For RNA-seq, I've seen some people go down to what's called a Q score, which we can see here, of like 20 when they are looking to trim. For genome sequencing, when I did genome sequencing, we actually wanted it a little bit higher, so we want all of our reads to have really high quality. It was set at 25 when I did it. Sometimes people go 27 or even 30. So if the sequence quality is below 30, it just gets thrown away because there's not as much confidence in it. So the sequence length, this is also something that's explained a little bit more down below, but our sequences for this one are 51 base pairs, and then our GC content is 47. And we'll also talk a little bit about that here in relation to the genome that we're working with, and how with RNA-seq it doesn't necessarily match up perfectly. But for DNA sequencing, if you did FastQC, this GC content should peak around the whole genome, what you would expect for the whole genome's GC content. So the first one after our base statistics is going to be our per base sequence quality. So remember before we had 51 base pairs is the whole sequence length. So here we have across the bottom each of our base pairs, or each of our bases. And then looking a little bit more at what's happening, the red line here is the median quality of those bases. The yellow box is going to signify the 25 and the 75 percentile. So 50% of the reads are between or are inside that yellow box. The whiskers, so on the top and the bottom, stretch from 25% to both 10%, the 10th percentile, and then 90th percentile. And then the blue, or it's really hard to see, but the blue line here, or black, if you can't tell that it's blue, that line shows the mean quality that's assigned for that base, so this base pair at this position. The green at the top is considered good quality or really good. So like I was saying, for DNA sequencing or genome sequencing, you want to have really good reads, really good base calls, so less uncertainty for it. And then for the orange, we have fair or decent quality, and then when you drop below 20, then you're starting to look at some trouble with more poor quality base calls. For this, we have a pass, so that's good. You can see most of our reads are pretty high. But a warning for this per base sequence quality will actually happen if the 25 percentile here, if a base call for the 25th percentile is less than 10. So we have 10 here. If any of these yellow boxes dropped below 10, we can see in this QC report, we have sequence quality, yep. So this one's 13, so not any of ours drop below, doesn't look like any of them drop below 10. So that's good. Or another thing is if this mean line drops below 25. So if the yellow box drops below 10, so you have a really wide range. Sometimes you see that with microbiome. So with the microbiome study that we did, this yellow box by the end was just stretching everywhere. Or we have the mean drop below 25, so about in here. So we have really good reads, which is great. A failure, so the red X, here will happen if this 25 percentile drops below 5, or if the median line, I should have said median, so this one drops below 25, or if the median line drops below 20. So if the orange line, not the blue, the orange line drops below 20, then we'll have a fail. So that's just something to keep in mind for the per base sequence quality. When we move on to per tile sequence quality, this gets into some of the information that the actual flow cell of the sequencer will provide. So can see if there is poor quality base calls. So again, we have 1 to 51. If we have poor quality base calls for a particular flow cell, so across we have our flow cells, and then we have our base calls vertically. And we can see here, like 1114, this one had a fair, no, it was kind of bad. The whole, for all of the sequence that was there, all base calls. The beginning wasn't too bad, but as it went down, it definitely did get worse. This will only show up for Illumina sequencers that keep track of the location of the sequences that are being read on that chip. So it's not something that's always going to show up. The more red means that the worse quality that base call was. So like we said here, the quality for base call 51, the last one of 1114 was more poor than good. And so the more red, the worse that quality was compared to the average of that column. So the average of all of these, the worse, the more red. And in some of them, I don't know if we have, we're looking for per tile. So it doesn't look like this keeps track of per tile, but sometimes you'll see just red, like deep red, like you see with the X across the whole thing. And that means that flow cell was just poor compared to the average of all the base calls at that location, but for all base calls. A warning here comes when a particular tile, so a row, has a quality score more than 2 less than that average for that column. So here, I'm guessing this is where our sequence quality warning, so the yellow exclamation mark is going to be that warning. So along here somewhere is probably the mark of where less than 2 of the average column comes into play. And then it's going to be a failure, so we'll have that red X if any particular tile has a quality score of more than 5 below. So whatever the column average is, if this one was more than 5 less than that column average, we would get a failure. Next, we have per sequence quality scores. So this is kind of what I was talking about before with the first one. You have your Q score, 27, 30, but we can see poor quality or good quality scores across all sequences. So this shows the distribution of those quality scores. So here, so this is rotated from the first graph. So we have our quality scores across the x-axis. Here's the quality scores along the y-axis. For all of the reads. So we can see most of the reads had a quality score that was rather high, so 38. Most of them, we could say it was probably greater than 30, which is really good. A warning here is going to happen when the average quality drops below 27. So if this was the average or less, we would see a warning for this. But a failure is going to happen if we have an average quality of 20 or less. So this is a cumulative reads. We have an average of 20 or less. We're going to have this be a failed quality check. Next, we have per base sequence content. So this shows the percent of each base that is an A, T, C, or a G. We can see generally we want A and T to have the same proportion or the same percentage, and we want C and G to have roughly the same percentage. If there are any like massive peaks or dips for one of them that isn't including this beginning part, that's probably an indication of an overrepresented sequence that is contaminating the library. So that could be from PCR or something like that if it's going to be an overrepresented sequence, and we'll get to some more of that below. And generally the beginning and sometimes the end are more poor quality, and they don't match up with the A and the T or the C and the G. Here, a warning happens if there is a difference between the A and the T or a C and the G greater than 10% at any position. So you can see here, we have a C way up here at 50% and a G here at 25, 26%. So since that's greater than a 10% difference, we have at least a warning, and a failure happens when that difference between the two bases is greater than 20%. So here, this is probably one of the locations where we're getting this failure. So if we have an A and T, it's not bad. Here is probably also another location where we're getting a fail, because the A is down at 18 something percent and the T is way up here at 40 some percent. So greater than 20, we have this failure. Next, we have our per sequence GC content. So this is what I had mentioned before with in a whole genome sequencing for DNA, we would expect this distribution to have a peak at whatever our genome GC content is. And the GC content of a mouse the mouse genome is about 41%, depending on where you look. Just quick Googles. And here we have, and we can see at the very beginning with the basic statistics, that our average GC content, this peak, is at 47. But this tells us the GC content per read. So we have our cumulative reads here. And across the bottom is that percentage. We expect a fairly normal distribution if the library is truly random. So very little only have 1 or 2 Gs, and also very few have all 51 be Gs or Cs. So very few or a lot of sequences have either none or all GC content. We're going to expect somewhere right around the middle, and that's where we get this peak. If we had a really long library, that say, no, the peak should be about the same regardless. If we had 23% GC content, we would see this peak here, and then it would drop off sharply and then go way out. So for RNA-seq, it doesn't always match up with what the genome content is, but for whole genome sequencing of DNA, it should be about correct. A warning here happens when the sum of the reads that's outside whatever this normal distribution is. The sum of the reads that doesn't match up with the normal distribution is greater than 15% of all of them. So here is the theoretical, what we should see if our peak is at 47. And since we're green, we're less than 15% deviated away from, or less than 50% of our reads deviate from that. And we're going to get a failure for our per sequence GC content if the deviation from that theoretical distribution is greater than 30%. So now we can move on to our per base N content. So at the very beginning I mentioned Sanger sequencing. For Sanger sequencing, generally the first 25, here it's only 50, so it's hard to see, but the first 25 reads are just bad, and the last 25 are bad. Thankfully for Illumina, it's generally pretty good all across. But this is where the sequencer cannot appropriately determine what that base is. So it'll just say N. There was a base here, but there's too much noise, or we just don't know what that base actually is. For this, a warning happens when the percentage of N's at any position. So here we have slight N content, but at any position, if the N content is greater than 5%, we're going to get a warning. And if we have a N content greater than 20% at any base location, then we're going to have a failure of our per base N content. And it just gets to be too many unknown. Now we can move on to our length distribution. Like I mentioned with with Illumina, generally it's pretty consistent. So here we have 0 sequences that are 50 base pairs. All of our sequences are 51 base pairs, and then none, 0, or very, very few are 52 base pairs. So that's good. Generally with Illumina, it's really consistent, which is great. But for PacBio, there can be a lot more deviation from just one. Like you can think some PacBio reads are 60,000 base pairs, but generally the average is a little bit lower, less than 10,000, between 1,000 and 10,000 are going to be most of the reads. And even nanopore, some nanopore reads can be hundreds of thousands of base pairs. So we might get a really long distribution tail for this as well. And a warning here is going to happen if not all the reads are the same length. So if we had some here, we would just get a warning. And then we're going to get a failure for sequence length distribution if one of the reads, one or more reads, have a 0 length. So if we have a 0 length read, it's obviously not a good thing. So that's going to be the sequence length distribution. Now sequence duplication levels. This is something that is a little confusing to me sometimes too, but so this blue line is going to represent the percent of the total sequences that are duplicated however many times throughout the whole library that we have. That's going to be based on the bottom. So we have about 50% of our total sequences only have one. So that's pretty low. Let's see if we can go here and find that too. Yeah, we can see a lot of them are at 1, but like the one that we have is somewhere in here. Anyway, this can be because of like a PCR bias. So if you have a sequence that is amplified a lot for PCR, that could be influencing this. But so for genome sequencing, we want most to be just 1, right? Because we don't have that difference that we would see in RNA-seq, where we have some cDNA in the library being more than others. So for RNA-seq, this could happen because we have some of those higher expressed mRNAs or lower expressed RNAs, which would show up here. So the higher expressed or the higher, more abundant mRNA may be here after PCR. So that could be something to pay attention to if you're getting these peaks for DNA sequencing, but for RNA, it's something to keep in mind, but kind of expected. Here, a warning is going to happen if a sequence is more than 0.1% of all sequences. So if one sequence is making up 1/1000th of the entire library, and it looks like we have something along that line. A failure here is going to happen if we have a single read or a sequence that is showing up more than 1% of the time. So if we have a sequence that's showing up more than 1 out of every 100, then we're going to have a failure for the sequence duplication levels. Next, we can move on to overrepresented sequences. And overrepresented sequences list the sequences that make up between 0.1% and more of that library. So up here we can see that we had a failure. So we had some sequences that were showing up at least 0.1% of the time, but not 1% of the time. And down here we can see those sequences. So we have 0.1% of the time up to 2.7. So this lines up with what we had for our sequence duplication levels. And it's possible that it identifies a source, but because it identifies a source does not mean that this sequence would have come from this contamination source. So we don't have any hit, so it makes it a little bit harder to explain. But what FastQC will do is compare this sequence against known, a small subset of known sequences to see whether or not it can find a possible source for why this sequence is now overrepresented. But the most we had is 0.27%, and the least that shows up here that's considered overrepresented is 0.1%. So like above. A warning is going to happen if we have sequences that are showing up 0.1% of the time, or 1 out of every 1,000. Or this is going to fail if we have sequences showing up more than 1% of the time. So if this one happened to be 1.27%, then our overrepresented sequences would fail, as well as our sequence duplication levels. So that's something to keep in mind. And finally, we have our adapter content. So this is looking for Illumina or whatever adapters that you're using to see whether or not they're present in the sequencing results that you have for this particular library. So this is 21.21.770 forward. So it's going to look for all of these adapters to see whether or not they're present at any base pair or any base call location. This gives you a good idea of whether or not you have to trim your sequences. I generally trim, or whether you have to adapter trim your sequences. I generally just put in the adapter trimming part, so that Illumina clip that we had done in the last video, I put that in anyway, just to be safe. If there's nothing there, it's not going to get rid of anything more, but it just gives me the peace of mind, I guess. And then this doesn't show it the best, and I don't have, so we don't have any, but this might be a little bit easier too. So at the very beginning here, we have 0.07, and at the end we have, so at the beginning we have 0.09, and at the end we have 0.11 and 0.12. So it increased. So why does that happen? Well, as soon as FastQC identifies a base call that has adapter, or it determines to have an adapter content to it, that's going to stick with that sequence. So if it finds it here, then if it doesn't find another location that has a adapter sequence in it, at the end, the least that it could have is what it started off with. But since it did find another location along the line that had adapter sequence content, it did go up. So in extreme cases, at the beginning, it'll start off probably relatively small, and as it gets closer to the end of the sequence, it'll increase, kind of like maybe a really slight S-curve. So that is kind of explaining the adapter content. A warning will happen with the adapter content if it is found that more than 5% of the reads have adapter sequences in them, and a failure will happen if adapters are found in more than 10% of the reads. So a warning is about here, and then it would fail if 10% of the reads had some kind of adapter content to them. So that'll do it for this video. Stick around, and maybe early next week or later this weekend, I'll post maybe the update for Trim-O-Matic, or potentially walk through what Trim-O-Matic, the code and all of that means, as well as some of the FastQC. We can do the MultiQC report too, and look at the results of trimming. So see you next time.